Hello friends, today we are going to discuss about microbiota. I am Dr. Suresh Badadmat, Professor of Psychiatry working at NIMHANS, Bangalore. In this video, I am going to discuss about the definition of gut microbiota, what are the functions of this microbiota, what are the psychiatric illness associated with microbiota, if there is a dysfunction of microbiota, how to be rectified, what are the practical implications, what are the current research and conclusions. All disease begins in the gut, Hippocrates said long back. Let's understand what is microbiota. The human gastrointestinal tract represents one of the largest interface between the host and the environmental factors. If you see the GIT, that is gastrointestinal tract, is hardly 5 to 6 meters. But however, because of the finger-like projections, villi, the surface area increases. The total surface area is approximately around 30 to 40 square meters. However, earlier it was considered to be 300 to 250 square meters. But now the research have showed that it is the surface area of half of the badminton court. Approximately 50 to 60 tons of food passes through this GI tract in a lifetime. That means this food will be digested, fermented, and utilized for energy. Hence, this gastrointestinal tract is considered as a bioreactor. Along with the food, when you consume, there are many threats. It may be bacteria, it may be virus, fungus, also goes through this GI tract. To neutralize this threat, there should be an immunity. That means there should be a defense mechanism that is called as microbiota. So microbiota not only ferments and digests the food, but also acts as a first line of defense against the attackers, that is bacteria and virus. Humans do not have any mechanisms to digest some of the food. So that means we require somebody else to help us in digesting. So these are the helpful bacteria or commensals bacteria which are there in the gut lumen. And they play a very important role in fermentation and digestion of the food. These collection of bacteria, archaea, eukarya, fungi and virus which colonizes the GI tract are termed as gut microbiota. There are 100 trillion microorganisms which are present in the human gut. These organisms have evolved, or I can say co-evolved along with the human beings over thousands of years to form an intricate and mutually beneficial relationship between the host, that is human, and the gut microbiota environment. They are called as commensals. That means the food we take is required for bacteria. They will ferment this and they will utilize part of it for the energy. Whatever it is left behind will be utilized by our human body for fermentation and then the digestion and usage for energy. So let's look into this diagram. You can see the GI tract starts from the mouth and ends at the anus. So here it is around 5 to 6 meter in length, but however, entire gut length, you can see there are 100 trillion microorganisms inside the gut. These organisms which are helpful may be bacteria, viruses, non-pathogenic fungi, and these play a very important role in fermentation of the food, digestion, metabolic, immune, and neurological function. And let me be very clear, 95% of the body microbiota is found in the gut. So this is how the microbiota plays a very important role. If you look at the ratio between the human cells and microbiota bacterial cells, it is 1:1. That means if you take the entire body cells in our body, and if you take the gut microbiota, it can be considered as 1:1. However, over 100 times the amount of genomic which is there in the bacteria and archaea content is more than the human genome. That means if we take the human genome and bacterial genome, it is 100 times more in the microbiota. The combination of both human genome and microbiome are called as metagenome. Let's understand what is this microbiome. Microbiome is the total genome content of the microbiota. So let's understand what this microbiota, where is it only in gut? No, it is also present in skin, airway, urogenital tract, eyes, and GI tract. These microorganisms play a crucial role in physiological functions, not only with regard to digestion, respiration, passing urine, and also in the eye. So these are commensals. If you look at the microbiota, there are 1,000 plus different species of bacteria in our gut. Each human requires around 50 to 60 grams of sugar per day to keep this microbiota going. And if you look at the 60% of the stool of human being has both dead or live bacteria. Microbiota is unique to the individual, but at the same time, we do share some of the microbiota among our colleagues, friends, family members within the same geography. Even the identical twins have a different microbiota. As I mentioned, gut is a bioreactor. Here, the food intake occurs, and by the help of this microbiota, fermentation of the food occurs. That means complex food particles are degraded by this bacteria. Once the fermentation occurs, further it is digested with various enzymes and it is absorbed for energy. Hence, gut is considered as a awesome bioreactor in our body. Once the fermentation occurs and energy is extracted, the byproduct is gas. Average per day, each human being produces approximately 2 to 4 liters of gas after the fermentation. And if you look at the total volume of the microbiota in the gut is, by volume it is 1.5 liters, by weight it is 2 to 3 kgs. That means microbiota, although we had not known for many years, now in the past 2 to 3 decades, the research has increased phenomenally. Let's understand whether the microbiota starts after the adult or by birth. Actually, microbiota starts from the day one till death. That means we can consider it as a life course perspective. The development of microbiota is generally believed to begin from the birth and continues throughout the life. After the birth or during the birth, when the child passes through normal vaginal delivery, the bacteria which are present in the vagina of the mother will be considered as one of the first microorganisms which are seeded in the microbiota. Similarly, if the child is born by C-section, the microbiota organisms are less. Also, further depending upon the breastfeeding or artificial feeding, and also what type of diet we take, and also the child is fed, on that basis also microbiota development is directly dependent. Further, various illnesses, diarrhea, medications, especially antibiotics, even a single dose of antibiotics can change the diversity of microbiota. Vaccination, stress, the way we share the food, environment, and being close and intimate with other person also play a role in the diversity of microbiota. By around 3 years of the child, the composition and the diversity of the microbiota is equivalent to adult. That means in a child who is 3-year-old, their microbiota is equivalent to an adult. The density and the composition of the microbiota are affected by chemical, that is fertilizers in the environment, nutrition, what we eat, environmental toxins, and immunological gradients along the gut play a crucial role in the development of microbiota. This diagram clearly talks about how the microbiota develops over the lifetime. If you look at the top one, prenatal and postnatal, there are hypotheses which says the microbiota starts developing as soon as the placental development occurs. That means mother transmits microbiota to the child through the placenta. This is during the prenatal. During the postnatal, it is through the delivery, through the breastfeeding, and also diet. If you look at the down, here the contribution is from the mother initially, later the food we take, and also in the environmental toxins, and also the food we take from the environment also play a role in the development of microbiota. Now, if you look at the diet, the breast milk or artificial milk, what are the solid foods, home, sharing of food, veg, vegetarian, non-vegetarian, individual diet, packed food, or else ready food, and also whether we are using the special diet, all these play a role in the development of microbiota. Further, if you look at the development of various illnesses, especially mental disorder, you can see in the second decade of life, that is, you can see if there is a disruption in the microbiota, there are studies which are indicating the development of depression, anxiety, eating disorder, drug abuse, bipolar disorder, schizophrenia, autism, Alzheimer's dementia, and Parkinsonism also been implicated. But however, with regard to brain function, memory, cognition, impulsivity, language, and social interactions play a role. Further, if you look at the gut, the development and the microbiota continuously occurs over a period of time. This microbiota also is essential for the development of immunity, regulation, and also filtering and keeping the immunity active. Further, as you see, the influential factors such as when the child was born, whether it is a C-section or vaginal delivery, what type of food is given, and whether the child was reared in the rural area, urban area, all those play a role. How often the antibiotics is given, and also if you look at the brain, and you can see various synaptic neurogenesis occurs over a period of time based upon the microbiota. This has been implicated and also been proved in the rodent lab studies. Now let's understand what are the functions of this microbiota. First and the foremost, microbiota plays a first line of defense. Hence, it maintains the integrity of the mucosal barrier in the gut. Further, it also helps in fermentation of the food and digestion of the food to extract energy. It also helps in providing various nutrients such as short chain fatty acids, vitamins, metabolites, neuroactive substances, gut hormones, and others. Food intake and craving to a particular person also dependent upon microbiota. And as I mentioned earlier, immunity. That means microbiota plays a crucial role in induction, training, modulating, regulating not only the local immunity in the gut, but also the systemic innate and adaptive immune response. That means GIT plays a very crucial role in development and training of the immunity. The interaction between the commensal microbiota and the mucosal immune system is very essential for proper immune function. Any change can also trigger autoimmune diseases in human being. Further, microbiota and brain axis is well known and there are studies pouring in. Further, gut and pulmonary axis, gut and cardio axis, gut and renal axis are also play very crucial role. Further, microbiota also plays a crucial role in learning, memory, and cognition through neurogenesis, neuroplasticity, neuroreceptor plasticity, and neurohormonal plasticity. Microbiota also been implicated how the drugs are metabolized and development of side effects in human being. So after discussing till now about the microbiota, let's understand certain definition and certain words which are there in this field. Microbiota. What does it mean? It is the community of microorganisms in the gut. They include bacteria, fungi, eukarya, and also archaea and viruses, and also few of non-pathogenic fungi. What is microbiome? The collective genome of the microbiota in the gut is called as microbiome. That means it is the genetic material of the microbiota. What is this microbiota diversity? It means it is a measure of how many different species and dependent on the diversity indices, how evenly distributed they are in the community. Lower diversity means that is dysbiosis. More the microbiota diversity means the person is healthy. So let's understand what is this dysbiosis. The opposite of microbiota is dysbiosis. That is altered gut bacterial composition, also considered as microbial imbalance or low diversity, has been associated with the pathogenesis of autoimmune disease and obesity in human being. That means dysbiosis means there is destruction or alteration or decreased diversity of microbiota. What is this colonocytes? They are epithelial cells of colon. They play a very crucial role in defense, immunity, and also in digestion of the food and production of various neurohormones and various other metabolites. Short chain fatty acids. Here, fatty acids with the 2 to 6 carbon atoms that are produced by bacterial fermentation of the dietary fibers. Without microbiota, human being will not be able to use this fat and also degrade them into short chain fatty acids, which are useful for us. That means microbiota plays a crucial role in production of short chain fatty acids. Let's understand some of the important terms. Probiotic. Probiotic are healthy or friendly bacteria that live inside our gut and support our health by converting fiber into compounds that have a benefits for our health. Probiotics means these are friendly bacteria. Now there is one more term called as prebiotic. These are group of nutrients, mainly fiber, that feed the beneficial bacteria in your gut. That means probiotics are bacteria. These bacteria needs a medium. That medium is fiber, that is prebiotics. Now there is another important term, postbiotics. These are bioactive compounds the probiotic bacteria produce when they consume fiber. That means probiotic is a friendly bacteria, prebiotic is a fiber. When bacteria acts on this fiber, produces postbiotics. And further, to add complexity, there is something called as synbiotics, which are a combination of probiotics and prebiotics, in which prebiotics are used to supplement, that is providing a source of fermentable fiber and enhance the viability of probiotic and postbiotics. So these are the terms which you need to remember. But however, you need to know about probiotic, prebiotic, and postbiotics. Let's understand the role of microbiota in lab. As I mentioned, in the past 2 to 3 decades, there has been various research occurring in this area. And this research play a crucial role to understand the role of microbiota in our human being. Let's look into that. The studies conducted to identify and examine the microbiota have been used different methodology. They play a very important role in understanding the complementary and also the intervention methods used in microbiota and how they are useful. The first and the foremost is germ-free rodents. I'll be talking about that shortly. Further, antibiotic-induced depletion. Here you give antibiotics and destroy the microbiota and then see by introducing certain bacteria whether it is helpful or not. Prebiotic and probiotic supplementation, whether is it helpful for human or not. Further, gastrointestinal infection and also fecal microbiota transplantation. These are the simple methodology have been used to understand the microbiota. The very important is germ-free rodents. Let's look into these germ-free rodents, are also called as mice. These are not but naturally colonized by microorganisms. Here, when the rodent or the mice is born, they are born not through a natural method. They have been removed by a cesarean section so that they are germ-free. And they are kept in a sterile environment. Sterile environment like this. That means the rodents are kept separately and they are handled in a sterile environment. And if you do any kind of culturing and to look for bacteria, there will be no bacteria yield on culture. That means they are completely germ-free. Now, by introducing various bacteria and see how these rodents grow or not. And these germ-free animals play a very crucial role in understanding whether microbiota is helpful or not. There are various studies have been done. The studies such as where a hyperactive rodent stool is utilized and introduced into the mice which is germ-free, and see whether after introduction of the gut microbiota of a mice which is very hyperactive, after a few days you can see germ-free mice also becomes hyperactive. Similarly, if you introduce a mice which is very silent and withdrawn, if you use those stool or the microbiota from that gut of the mice to a germ-free mice, and you can see that within a few days the germ-free rodent becomes silent. So these kinds of studies are occurring day in and day out. Now, they have studied these germ-free mice after the microbiota has been introduced. They have found there are differences with regard to germ-free mice and also in normally fed mice. In germ-free mice, they have found that their gut is leaky. That means the pathogens can grow easily into various parts of the body. That means it is highly vulnerable for infection. They do not have good immune system. Their immune system is very poor and very weak. They do not able to digest. They are generally weak. Nutritional deficiency is characteristically seen, and also poor functionality is seen in these mice. There is decreased learning, poor motivation, poor memory, and also cognitive functions are very less. Further, lack of socialization can be seen in germ-free mice. And there are significant changes seen in these germ-free mice, such as decreased neurogenesis, decreased microglial activity, decreased synaptic and neuroplasticity, decreased myelination, and also myelin sheath, and also decreased brain-derived neurofactor. Blood-brain permeability is very high, gut permeability is very high, and also decreased hippocampus and amygdala volume, which indicates there is a decreased memory, decreased learning, and also decreased neuroplasticity in these areas. Let's understand what are the research on microbiota. With the advent of culture independent approach, that means you need not have a culture to understand these bacteria. Culture independent approaches to identify the microbiota targeting the bacteria, that is the bacteria's 16S ribosomal RNA. This RNA gene sequencing helped us to identify. In this 16S ribosomal RNA gene is present in all bacteria and archaea and contains 9 highly variable regions, that is V1 to V9, which allows us to identify the species and also able to know what is the strain which it belongs to. The utilization of this shorter read length can also add to the error. Hence, there was a further refinement in the technology. To understand this, the composition and diversity of microbiota may be provided by whole genome shotgun metagenomics. Due to the higher resolution and sensitivity of this technique has been utilized. It means from 16S ribosomal gene and further refinement into whole gene shotgun metagenomics have been utilized to understand the microbiota. And various important research projects have occurred, such as MetaHit, Human Microgen project also has been considered. Further, Microbiome project also are very important to understand the microbiota. The results from these projects, the compiled data from these projects identified 2,172 species were isolated from human beings and classified into 12 different phyla. In that, 386 species are strictly anaerobic in nature. So, these have been identified and subtyped. Further, an extensive catalogue of the functional capacity of the human gut microbiome was recently obtained, where 99,87,896 genes were identified through a combination of 249 newly sequenced, and you can find 10,018 published samples. The study identified the presence of country-specific microbial signatures, suggesting that gut microbiota composition even it is shaped by the environment factors such as diet and also the host genetic factor. For example, if you want to study the microbiota in a geographically well-defined location, there may be sharing of microbiota. That means you can see the microbial signature from specific place depending upon what food they eat, what is the environment, how much fertilizer is used, how much organic fertilizers is used. All this plays a role in the development of microbiota and also maintenance over a period of lifetime. Yes, microbiota is not constant. They're continuously changing. Gut microbiota is continuously changing in the gut environment. Ecosystem of the bacteria in the human gut operates dynamically. It depends directly upon the lifestyle, what is the physiological state of the person, what food intake, stress, smoking, alcohol intake, antibiotic intake, genetic vulnerability, from which place he is coming, geography, age, gender, resilience, the type of birth, and also various other factors plays a role in this microbiota, and it changes every moment and every second. Further, the interaction of the microbiota and diet also plays a role in the development of the microbiota. Further, this microbiota ferments the food, and that smaller part of the particles will be considered in the protein and the development of genes. And these genes and also those broken down food particles can be considered as a metabolite of microbiota. Let's understand microbiota histologically. The mucus is present throughout the gut, that is GI tract, and it is thickest in the colon, where it is crucial in mediating the host microbiota relationship. If you look at the colon mucus, it is divided into two layers: a dense, impermeable, sterile inner layer, a loose outer coating which is penetrable by bacteria, and energy source. Here, the preferential binding site for commensal bacteria occurs. Let me repeat. The mucosal of the colon has two important layers: a dense, impermeable inner layer which is sterile. The outer layer is loose and it is highly penetrable. And here, the commensal bacteria are attached and the energy source starts occurring here. If you look at this diagram, you can see the gut where the microbes are there, where microbiota. From there, after the, you can see that clearly the genes and proteins are formed after the fermentation and digestion, and further it is produced is the metabolite. You can see the bottom-up approach and also top-down approach. First, as I mentioned, that is the gut microbiota, where you can see in the bottom-up approach, various bacteria are there and also various fungi and various other microorganisms. This will degrade the food or the ferment the food further, and there they produce various genes and proteins, and further it is degraded into various metabolites. In the top you can see the healthy gut, that is, there is no inflammation and you can see the thick mucus. But however, as you go down here, the mucus is reduced. That means the mucus is not there and the gut has been compromised. Here, the cells, pathogens can easily enter into the gut, that is basically into the wall of the gut, and producing inflammation and causing various illnesses in the host. Further, as I mentioned earlier, there are various metabolites produced from the microbiota. The gut microbiota provides essential capacities for the fermentation of non-digestible substrates like dietary fiber and endogenous intestinal mucus. That means some of the fibers we do not have a capacity to digest. Microbiota helps them. This fermentation by the microbiota supports the growth of specialist microbes that produces short chain fatty acids, metabolites, and other gases. Here we are very interested in short chain fatty acids. These short chain fatty acids is very important nutrient for our body. The major part of the short chain fatty acids are butyrate, acetate, propionate, and various other metabolites. Here we will discuss each one of these: butyrate, acetate, and propionate. Butyrate. It is the main energy source for human colonocytes. Can induce apoptosis of colon cancer cells. That means the butyrate plays a very crucial role in protecting against cancer. Further, it also plays a crucial role in the intestinal gluconeogenesis. That means in extracting energy. It also plays a crucial role of hemostasis through beta oxidation, generating a state of hypoxia that maintains oxygen balance in the gut and preventing the gut microbial dysbiosis. That means butyrate also indirectly helps microbiota's environment. Let's understand about the acetate. Here, it is the most abundant short chain fatty acid, an essential metabolite for the growth of other bacteria. Here, this acetate helps in the growth of other bacteria. It is used in the cholesterol metabolism and lipogenesis, and that means 15 to 20% of the energy is produced here. May play a role in the central appetite regulation. The acetate, once you take the food, and food is fermented in the microbiota, and further acetate is formed. This acetate may play a role in giving feedback to the hypothalamus: yes, this person has taken food and the appetite is suppressed. Now let's understand about propionate. Propionate, once it is formed in the gut, it is transferred to the liver, where it regulates gluconeogenesis and satisfies the signaling, satiety signaling, through interaction with the gut fatty acid receptors. And butyrate and propionate control the gut hormones. Further, other products are also formed in the gut, such as trimethylamine. This is a toxic substance which helps in development of atherosclerosis. That means we need to decrease trimethylamine formation. If the atherosclerosis is developed, as you know, the artery pliability becomes very less and hypertension develops. Similarly, there is indole propionic acid development. This also causes diabetes too. That means there are some metabolite which should not occur. Whenever there is a dysbiosis or the microbiota changes rapidly, and because of the food we take or fatty foods we take, or else the cooked food, or it may be ready food which is available on the shelf if we take, may produce this trimethylamine or indole propionic acid. They play a important role in the development of these illnesses. There are various other metabolites are being studied, such as gut hormones, neurohormones, neuroactive substances, liposaccharide, polysaccharide, peptides, enzymes, glycoprotein, microbiome, that is, nervous system of the gut, pro-inflammatory cytokines, ghrelin, and 5-HT plays a crucial role, and other metabolites. At this point of time, we know few of these. There are many substances we are unable to code at this point of time because research is currently in progression. Let's understand about dysbiotic. As I mentioned earlier, dysbiotic means decreased microbiota, or else altered microbiota, decreased diversity of microbiota, or else decreased density of microbiota. Here, these altered microbiota is not helpful for human being. This dysbiotic microbiota might be the one which fails to provide the host with the full complement of beneficial properties. That means the products which are formed after fermentation and digestion is harmful to our body rather than providing energy. Here, there may be reduced diversity of the microbiota, decreased number of commensals or helpful bacteria, and there may be pathogenic microbiota. That means harmful bacteria can start growing. And a plethora of diseases have been implicated to dysbiotic, from development of neurodevelopmental disorder to psychiatric disorders. If you look at this diagram, on the right side you'll be able to see there is a green color which talks about the helpful microbiota, where there is a dietary intake is there, probiotic intake is there, increased short chain fatty acid, increased antioxidant, decreased indoxyl sulfate, which is kidney toxin, further improved lipid metabolism, low gut inflammation, insulin sensitivity, and reduced risk for infection. That means on one hand, you can see the health. On the other hand, you can see there is a disease. What are those diseases? At the end, you can see there is altered pH in the gut, which leads to excessive protein consumption and also excessive sugar intake, excessive saturated fatty intake, proton inhibitors, antibiotics, which play a crucial role, and short chain fatty acids are decreased, and also various other changes occurs, which leads to various illnesses like hypertension, diabetes, cognitive decline, and neurodevelopmental disorder. So that means the early or preliminary studies indicates microbiota plays a crucial role in various illnesses, not only from the brain, it also in the pulmonary, and also with regard to systemic hypertension, diabetes, endocrinal system, renal system, colon system. That means microbiota cuts across various departments of medical fraternity. Thank you very much for giving your valuable time. Stay safe.